One of the manifestations of metabolic derangements in cells is the intracellular accumulation of substance that may be harmless or cause further injury. These accumulations may be located in the cytoplasm, within organelles, or within the nucleus, and they may be composed of substances that are synthesized by the affected cells or are produced elsewhere. Stockpiled substances can be normal cellular constituents such as water, lipids, proteins, or carbohydrate, abnormal substances such as minerals or products of infectious agents, and pigments. There are four main mechanisms leading to an abnormal intracellular accumulation. First one is inadequate removal of a normal substance, secondary to defects in packaging and transport, as in fatty change in the liver. Number two is accumulation of endogenous substance as a result of genetic or acquired defects in its folding, packaging, transport, or secretion, as with certain mutated forms of alpha-1 antitrypsin. Three is failure to degrade a metabolite due to inherited enzyme deficiencies, and this is seen in lysosomal storage diseases. And number four is deposition and accumulation of abnormal exogenous substance that cannot be degraded by the enzymes in the cells, and it accumulates, such as in carbon or silica. All major classes of lipids can accumulate in cells: triglycerides, cholesterol, cholesterol esters, and phospholipids. Fatty change is often seen in the liver because it is a major organ involved in fat metabolism. As you can see here, this is the picture of a liver, and you can see fat is deposited within the cells of the liver. So another important example is atherosclerosis, where lipid is deposited in the intimal layer of the blood vessel. In disorders with heavy protein leakage across the glomerulus, there is increased reabsorption of protein into vesicles, and this protein will appear pink within the cytoplasm of the tubular cells. You can see all these pinkish granules. Those are protein that has been reabsorbed in the renal tubular epithelium. Pigments are colored substances. Some are normal constituents of the cells, such as melanin, and others are abnormal and accumulate in cells only under special circumstances. Pigments can be exogenous, coming from outside of the body, or endogenous, synthesized within the body itself. One example of exogenous pigment is tattooing, which is a form of localized exogenous pigmentation of the skin. So the pigments inoculated are phagocytosed by the macrophage in the skin. Another is carbon or coal dust that are inhaled in people living in the urban areas. So they accumulate as black pigment in the lung and the lymph nodes. Fusin is an example of an endogenous pigment, and it is an insoluble pigment that is not injurious to the cell or its function. It's a wear and tear pigment, and you can see those brownish granules within the cardiac myocyte. Another endogenous pigment is hemosiderin, which is a hemoglobin-derived golden yellow to brown, granular, or crystalline pigment. Okay, and you can see it here, these brownish pigments within the liver, and they are representing aggregates of ferritin micelles. So you can see this is a special stain for iron. You can see the iron pigment or hemosiderin is blue in color. So for this session, we have described the differences between the types of adaptations. We have explained the causes and mechanism of cell injury. We have described the changes in reversible cell injury, and we have also described the different types of intracellular accumulation.